thanks. We'll see if I can figure out the technology of the slide projector, because that's really complicated compared to what we do in the lab, right? So, at any rate, I'm really happy to be at the Nutrition Research Institute. My group moved here about a year ago from where we were working in the Research Triangle Park. And we're really fortunate to have a very nice state-of-the-art lab in this building. And our laboratory is up on this top, and we have lots of really interesting kind of equipment, and I'll show you some pictures of that in a little bit. And then we have a lovely office suite here, and you're welcome to come visit us sometime if you'd like to get a tour of the labs and see what we do there. We'd certainly like to have people come and see what we're doing here. So the kind of work that I do, I refer to it as biochemical fingerprints. And we take these biochemical fingerprints to try to understand our health and wellness. And I'll tell you a bit how we do biochemical fingerprints. But it's sort of like taking a handprint where everybody has a fingerprint or handprints that are unique. We have uniqueness in our eye colors and in our skin tones, and we can put these various things together and look at somebody and define their look, their characteristics. And then what we do is understand all these things that are biomolecules that are present in your biological fluids and tissues. And there's thousands of those biomolecules. So it's very challenging to be able to measure them. So we have to have really good people like Yon Lee, who knows how to go in and measure all these biomolecules for us. Okay, so as Steve mentioned, we were very fortunate early on to get funded by the National Institutes of Health to set up these technologies for measuring instead of just a few metabolites at a time, which you may get if you're going to the doctor and they're taking your blood or urine samples. We can now measure thousands of signals simultaneously. And this is what we call metabolomics. And there's 6 cores in the US, and our core here is now at UNC, and there's one at Michigan, UC Davis, one at the Mayo Clinic, University of Florida, University of Kentucky, and then all of our data that we're acquiring for people all over the U.S. and even samples coming from outside of the U.S. goes into a coordinating center. And so we can get this data for thousands of signals from all these different people and start seeing what it looks like based on where you come from and what you've eaten and what you've been exposed to, right? So that's kind of what we're doing. Okay, so one of the things that we want to do at NRI is really understand what our biochemical fingerprint has to do with our genetics, or maybe it's what does our genetics have to do with our biochemical fingerprints. So we want to understand that individuality in genetics and then how that relates to our disease. So some of us know our genetics pretty well because of oral history in our families, and I'm from around here, so I think I might be the only scientist on our staff that's from around here. So I have folks from Troy and Albemarle, Mount Gilead, and Midland, and Oakborough. So I actually know these names, so you know I'm from around here then, right? Because otherwise you just wouldn't even know these names. From Concord, and then from Stanley County, Frog Pond, Union, Red Cross, all those good places down there. And so we can know about our genetics through knowing what we've been told and who we've been told we're related to. And then also finding out about, you know, that sitting on the porch, talking to all our relatives, people that aren't relatives, trying to figure out how we are related. It's always a lot of fun. Oh, my husband's calling, trying to figure out where I am. Okay, turn that off. Okay, and then there's also other tests like 23andMe and Ancestry, where you can submit your DNA samples and then start learning more about your DNA and find out people that you are related to through those DNA databases. I think I have 1,700 cousins in my DNA test, which is kind of cool. And some of them I figured out how I'm related to because of the oral history, some not yet. And then there's other ones like Ancestry. So what we want to do is really understand people's DNA, what's going on in their genes and their genotype, and then how that corresponds to these biochemical processes that I've been talking about that we often call the metabotype. Okay, so that's how we get to the metabotype, right? All right. So, and how those are related, you probably remember this from like 8th grade science, but I'll say it anyway, is that, we all have our DNA, and that contains our genetic instructions, and it's responsible for making proteins, and those proteins are responsible for making metabolites. And so if things occur like exposures or diseases that we have, a lot of times those will modify our proteins, and they'll do something that's called a post, right after the protein, post-translational modification. And then that affects the types of metabolites and how many of those metabolites that we have in our body. Okay, and then we also can have genetic mismatching or things that happen at the chromosome that can also make a difference in what one would have in their metabotype. So there could be either the direct or this indirect through the protein of how it changes our metabolism. So, but even if you're healthy and you don't know of any diseases that you may have, you're still going to have a unique metabotype because we have unique genetics, right? So our metabotypes are unique. So we're going to have a test on this at the end. So I'm going to start up here in this corner, and I'm going to read off all the metabolite names, and then we'll see how many you can remember, right? Because we're going to include that in one of our memory testing studies at NRI. No, just kidding. So what we do is we know a lot about biochemistry. Biochemistry's been studied for a really long time. There's lots of journal articles that tell us about what metabolites people have inside their body and how those are connected together in pathways. So what we're doing that's different is we're trying to study all of those at one time. Very challenging. Okay. So now how can we do this? So what's that have to do with what's inside your body? Okay. So you have DNA, right? And that does your genetics, right? But all of your cells and tissues and biological fluids have lots and lots of metabolites. And they're in your serum, which is from your blood, right? Plasma, which is from blood, in the urine, the saliva, in things like cerebral spinal fluid, of course, comes out of your spine. And they're also in your organ tissue. So any organ tissue in your body, if you were to grind it up, you have lots of metabolites in it, right? So that's what we're trying to measure, are all of these things that are going on. Whoops, don't know how to go back because technology limited. No, okay, got it. Okay, so all of these things, we like to use these kind of serum and urine and saliva and breath and plasma because they're what we call relatively non-invasive. So when we're doing studies that people want to participate in, these kind of samples plus stool samples are things that people can give us without us having to do damage to their body to get a tissue, right? And so by understanding what's going on in these, we can tell about the health of the organs that are inside the body, right? And of course, at our institute, we really want to try to understand that health in regards to nutrition. Yeah. So, so one thing that we so to put it in the context of your doctor's office visits, I was saying that we all have like a unique metabotype. So that's going to depend on our gender, our age, our ancestry, and in our health status, right? So you can be well or you can be sick, and your metabotype's going to change. And you kind of know this because when you go to LabCorp, right, and you give a urine sample or a blood sample, you get the labs back, and it tells you whether or not your glucose is above normal, right? That would be a normal one. Or your sodium is above normal or below normal, right? I think we just usually hear about the above things, right? And then there's enzymes, like maybe if you've had any kind of liver injury, you may find out that an enzyme called an ALT is elevated. So those are the kind of things that we get from LabCorp. And those kind of test results are really great, but they tell us a lot about when we're sick instead of how to stay well. So we're trying to move more towards the early diagnosis before you get sick to tell when you're deviating from wellness. And what could you do to change that process when you start to deviate? Okay, so what Metabolomics is bringing now, so we started into Metabolomics in about 2000, and our group was very early into using this approach, creating the approach and refining it. And it's still not perfect, of course. It takes a long time. But what Metabolomics is bringing now to the literature, which now influences what's going to be adopted in the clinic down the road, are more sensitive and early markers for disease, which is something that's really needed. So we don't want to wait until we're sick to find out what's gone wrong. Okay, and then also markers to monitor treatment. So that could be if you're taking a drug, we want to know, is that drug going to work well in you, right? Or is it going to work well in someone else? So we want to be able to understand your signature and how a drug treatment will work or a nutritional treatment would work. So some people who are on, say, one diet might not lose weight while other people may lose weight. So those are the kind of questions we can ask, and we can say, are there things about your biochemical profile before you start a treatment that could inform your clinician or your nutritionalist what your outcome is going to be. Okay, so you're going to have a good outcome or a good response or not. Okay, so what we're doing is moving away from using just these clinical measures that I showed from like the LabCorp kind of measurements to doing things you may have heard of, protein expression or proteomics, which I don't do, but some of my colleagues do, and then this metabolite data. And we look at what we call these metabolic pathways, where we look at the interactions of all these metabolites and try to look at the focal point of why someone is deviating from wellness. So what metabolomics is then, we're measuring all these low molecular weight compounds in cells, tissues, and biological fluids. And then we're trying to determine how to uniquely profile your biochemistry unique from anyone else's biochemistry, and then to monitor that over time to understand the deviation from wellness. So that's kind of the thing. Okay, so what we really do every day is, we do a lot of things in the laboratory, and I'll just tell you a little bit about that. We do a lot of sample preparation of these biological specimens, and we get those from human subject studies, the kind of study that Katie was talking about earlier, where she'll be getting samples of plasma and stool samples, and then we can analyze those with metabolomics technologies and start looking at what are our signatures of wellness and deviation from wellness. And we worry about things that everybody's always worried about in any kind of industrial setting, like calibration of all of our materials that we use to run the assays. In this case, the picture's for a pipette. So no matter sort of what kind of occupation you're in, the same kind of things we worry about. We have quite a bit of equipment in our laboratory. Here's some of the staff that's currently working in my lab, and then some former staff. We use mass spectrometry equipment, which allows us to do kind of what it says, look at the mass of a molecule. So if there's thousands of molecules in the sample and they go into this instrument, they do something that's called ionize. So at any rate, they kind of get a charge, they get fired up, right? And then they hit our detector and tell us about their mass. And the time it takes for them to hit the detector, or the strength in which they hit the detector, can tell us about how many of those molecules there are and the structure of the molecules, okay? And so we do that. You can see various types of equipment here that we have within our core lab. And so we spend a lot of time running standards, which would make a lot of sense if you're trying to figure out what you're measuring. You have to have some sort of standard to run against. Doing quality control on all of our sample preparation and all our instrumentation. Checking reproducibility. There's a lot of auto-sampling that we have set up now, so it doesn't take a human to inject every sample, but we can put those on these auto-samplers. There's a lot of cleaning and tuning that goes on with the instrument, so you have to have a specialty in this mass spectroscopy to do that. And then there's a lot of data analysis. And because now we have these thousands of signals, the data analysis is really complicated. And so there's a lot of statistical and multivariate analysis techniques that are used to try to unravel all of these peaks and to line them up with metabolites and which ones are telling us about wellness and sickness, right? And we also collaborate with people that are at the DHMRI facility that you probably know about, the core lab. And so we do also some collaborations there as well as in our group. So I sort of like this slide because years ago when I was in high school, I worked in a mill right outside of Gastonia. It was called Threads. Has anybody heard of that mill? I spun thread, so it's kind of cool. I did, and I really learned a lot from spinning thread that I feel has been important to me in my job now, because I learned about cleaning and tuning and running standards and quality controls and calibrations and, you know, even making everything automated. So it's really kind of cool how that experience, you know, I think was one experience that got me really interested in science and machines and doing things like that. So I just thought I'd add that in because it's really nice to be working back here now. So anyway, so what's metabotyping told us with these thousands of signals? Well, I think of metabotyping as being the center of the universe because that's what I do. Most scientists think of what they do as the center of the universe. So that's what's drawn here in the middle is metabotyping. And everything you do influences your metabotype. So, well, you got your genes when you're born. They influence your metabotype. But then what you, drugs you take, any kind of drugs or dietary supplements, or even if you're exposed to things in the environment or whatever plants you eat or foods you eat, those all influence your metabotype. And then other kind of environments do that, like if you're stressed out or if you're really happy, or what do the other ones say? Oh, lifestyle factors like if you're smoking or not smoking, or if you're jogging or not jogging. All of these things change what's going on in your metabotype. So if you look in the literature, you will see since the year 2000, and in science between 2000 and 2018 is not that long, right? That studies have gone from pretty much zilch in the area of metabolomics to a very populated literature that shows that our metabotype correlates with all of these factors that I was just talking about, plus many health factors like blood pressure and weight status and mental health and many types of diseases and behaviors. So it's kind of cool. So, but there's so many things that affect our metabotype that makes it really, really complicated also. So we have to design very careful studies to try to figure out what metabolites are important for that specific disease, right? So I'm going to tell you about a few studies now, okay? unless somebody wants to ask me about spinning thread or what color I did, or I made my way up to white, and I want everybody to know that because that was a big deal. Because yeah, so I did. I made my way up to white. Anybody who's spun thread in a mill knows what that means, right? So anyway, so this is one study that we did recently, and we collaborated with Michelle Williams, who's shown here, and one of who was an intern at the time and now is a professor at Harvard, Bazou Galay. And this study was on placental abruption. And placental abruption is an ischemic placental disorder that results from premature separation of the placenta before delivery. And it's believed to occur in about 1% of pregnancies, or at least 1% of pregnancies report that. It might occur at higher rates, but it's at least 1%. It's associated with preterm delivery and fetal death and hemorrhaging and renal failure. It's incredibly difficult to diagnose because early on during pregnancy to the obstetrician, the women can present as normal. So they go in for their visit with the obstetrician and everything looks fine. And typically, there's no signs that somebody's going to have a placental abruption until it happens, right? So what we wanted to do was a study to see if we could find biomarkers in the first trimester of a woman's pregnancy that would predict the probability of that mother having a placental abruption. And so Michelle is an epidemiologist at Harvard, and she's actually chair of the School of Public Health, Dean of the School of Public Health there. And she has a large biorepository of samples from women. It's called a pregnancy cohort because there's been these samples collected over many, many years from pregnant women. And they start collecting them at the first time that the woman goes to the doctor and they know she's pregnant, and at that time there's no signs of any complication, and they continue to collect them throughout pregnancy. And some of those women have an adverse pregnancy event, right? And some of the women are fine, right, at the end. So she selected samples from her biorepository of what we call cases and controls. They're people who either did have a placental abruption or didn't. But it's important to know that it was in the first trimester, so there were no signs to the doctor that the woman was sick, okay? And so what we were able to do is not find a lot of metabolites, but 9 metabolites that were associated with having a placental abruption. So this was from the first trimester serum. And so we could look at those names of these metabolites and go, well, what does this mean? Could we do anything about that? Well, it could be a biomarker that a clinician could use to be able to predict whether somebody's going to have a placental abruption or not. But is there anything else we can do with that information? That's what we do, right? So after a lot of complicated mathematical modeling, that's all I'm going to say about that, unless there's somebody who wants to test on this slide, and we can go over it in detail. Okay, we were able to find out that our metabolites were more predictive of having a placental abruption than what the doctor currently can measure. So we're really happy about that. So that's called beating the gold standard of the clinic, right? At least in my world, since I'm a chemist, right? So I think we did better than the doctor could do. So, you know, that's pretty cool, right? And they think that's cool also. But we do mathematical modeling to prove that, and that paper was published recently. And then we try to understand then, well, what does that mean in terms of people's biochemistry, right? So what we found is that the probability of having a placental abruption was increased in people who had an increase in what's called acylcarnitines and a decrease in phosphatidylcholines. So this takes us to this pathway. So in that big complicated pathway I showed at the beginning where you couldn't read anything, now we're down at some little minuscule amount of that, right? So we get down to this pathway, which I'll refer to as the choline pathway. And this compound, phosphatidylcholine, is related to this pathway, and this is what was decreased in people that had a placental abruption. So, you know, to me, I'm a chemist, it seems pretty easy that choline is definitely it's known to play a critical role in pregnancy and in women's health and neurodevelopment in babies. So it seems like, well, maybe we would increase choline in the diet or phosphatidylcholine in the diet, right? And perhaps that would be a nutritional intervention for placental abruption. So, of course, we can't go and do something like that until we validate the study. So right now we have 230 samples that are being analyzed in what we call a larger cohort study to see if we can get the same results again. So that's the next step, right? But choline is something that we can get in our diets through eating eggs. And so eat your eggs. Don't overeat your eggs. I'm not an MD. I'm not going to tell you how many eggs to eat. But I will say yeah, thanks. Yeah, I always have to give that disclaimer. I usually eat 2 in the morning. But Dr. Zeisel, who's our director at NRI, has spent decades working in choline metabolism and has many, many publications on the benefits of choline in the diet and the understanding of what the adequate intake is for women and people, but particularly women and even women during pregnancy. And many people have below the adequate intake of choline. So it is something that you could possibly want to talk to your doctors about, right? This is also related to this other pathway that I have listed here that's very closely related, which is folic acid. And many of you probably know about folic acid because you know that during pregnancy women get a supplement that has that folate in it to prevent spina bifida, right? And Sergei Kropinko, who's here, is one of the world's expert in folate metabolism and can tell you anything you want to know about that, right, Sergei? He's a biochemist, but he can still tell you, right? He said okay, he would. Okay, so that's an example of sort of what I do, and you know, I have to admit, I don't really solve the problem because that's, you know, we do what we do and then we inform the next step, right? And then they look at that. People like Katie may look at that, who's an epidemiologist, to decide, well, what are we going to do with this information? So that's one kind of workflow that happens, right? So then I'll tell you a little about another study. I think I had put on the thing I'd talk about a few studies, so I want to live up to that. So autism is another area that we've worked in. And I think we were the first group to publish a paper using metabolomics to study autism. This was done with samples from children that are in China. It's a Chinese Han population. And this was 3 to 6 year olds. Half of our subjects were autistic and half of them weren't autistic. One of the reasons why the Chinese Han population was so ideal for this study is that the children are not on medications already, because autism in that population is just like, well, that's how you are, you know, and this is how I am, and you're not, you know, we're not putting you on medication, you know, because of that, you know, at least in this group of kids. So they're not on medication. What we were able to do was we discovered 17 metabolites that we could identify in our first study as being linked with the autism children, different from those who didn't have autism. And we did a second study where we validated 11 of those 17 metabolites. So the point is, we don't always validate everything, but when we do validate, that's really cool. So of those things we validated, there were 2 compounds here that had known links in the literature to what's called a sphingomyelin. People knew that that was related in subjects with autism spectrum disorders. And then another one that was really interesting is DHA. This DHA actually in the United States is given to many children who are autistic. And in our study, we saw it at lower levels in the autism population. Then there was a list of other analytes that we were able to validate, and these are here. So the question is, what do we do with that information, right? So you're not going to run and start giving that to children to see if it makes them feel better or not. So that's not how we do it. The next step of the workflow is colleagues who have animal models that have behaviors that are autism-like are now introducing these different compounds into the animal models to see if it reduces some of the autism-like behaviors, some of the side effects that are associated with autism. So once we understand how that works in animal models, then we'll know more about what we may want to do in humans. So we'll test it in the animal models first. So that's another kind of workflow that we have. So I was saying earlier that we know that our cells have all these metabolites in them to map to those biochemical pathways. But you must also know, and I hate to tell you this if you didn't know this before, that you also have a lot of things in your blood and your urine that you wish weren't there that's coming from exposures that you have. You could have a workplace exposure. Like when I worked in the mill, when I was a teenager, no one told me about exposure, right? So I probably have exposures from that. And when I was a child and would help my dad paint, you know, we would paint houses and wash out paint brushes and things. I'm sure I got a lot of exposures from those chemicals also. I'm probably a mess biologically. And then also, so there's things that you may have in your home or you may use in your workplace. And then there's other just pollution, car pollution, right? Standing on the side of the road, being by a factory, right? So you get these chemicals and they're in your blood and urine. And this is, Jan mentioned you before, there you I'll mention you again. She's been setting up this really unique way of being able to measure what we call the exposome. And this includes not only those good endogenous chemicals that we want to be in there, but it also includes the measurement of a lot of compounds that may be in your blood or may be in your urine, but we really don't want those in your body, right? And there are things like that come from pesticide exposure would be an example, okay? Or from drinking, remember all the big thing about drinking out of certain water bottles or babies getting exposed to BPA? Those kind of things, right? So you read about those in the news. So what happened is all that news got to our federal government, and so they started thinking, well, how are we going to take care of this problem? How are we really going to understand this? How are we going to understand how all these chemicals affect particularly children? Because we're, you know, this vulnerable stage where your organs are developing and everything. How do these exposures influence that? So National Institute of Environmental Health Sciences, which is in North Carolina, right? And I think it's the only National Institutes of Health institute, which is not located in, I'll say, DC/Maryland, right? So we're really proud that North Carolina actually was selected to have this here. This is in the Research Triangle Park, and they fund research like we do, and they've funded us to be a children's health exposure analysis resource hub. So that's where part of our money comes from. So usually scientists do research on whatever they get paid to do, right? So that's where part of our research funding comes from. And what we're trying to do is to expand the use of environmental exposure analysis in children's studies or in women's health studies, okay? So that's cool. So now one of the things that happened to make NIEHS decide that they wanted to do this was literature started coming up on the use of metabolomics in animal models that were model systems that would make one concerned about what would be happening in humans. And one of those studies came out of my lab in 2009, okay, so before they had decided to start these kind of cores, and one of the reasons why we got funded. And what we did is we took pregnant rats and exposed them to what's called butylbenzoyl phthalate. It's compound that's ubiquitous in our environment, and you may be getting it in your water now, not to scare anybody, but whatever. But you can get that phthalates from a lot of sources in your foods. And so we wanted to see that if we took some pregnant rats and exposed them to the phthalates and other pregnant rats and didn't expose them, would the offspring, the pups, the rat pups, have differences in their biochemical profiles? So I'll cut to the chase at the end of the story and say, at the end of the story, we could tell pups that were born to the mothers that were the control mothers, and they were different from the pups that were born to the mothers that got the butyl-benzyl phthalate. And so this let us start knowing that we can really use metabolomics to try to understand what's going on with in utero exposures, and then to associate that with health outcomes in the offspring later in life. If anybody wants to know the details, you can read this paper and stop by the lab and ask us about that if you'd like, right? Okay, so we did that with, and you've all heard of Krebs cycle, right? So we found like perturbations in the Krebs cycle, right, with the TCA cycle, and in metabolism of your amino acids and, tryptophan, so we could tell that there was an impact on metabolism. And we've done other studies now, either in animal models or in humans, with other environmentally relevant compounds, like arsenic, that sometimes is in drinking water. We published a paper on that recently in a pregnancy cohort study. Or we're doing some work with nanoparticles in a rodent study. And nanoparticles are becoming really important. It's something that I'm really proud of our federal government. They're trying to get ahead of understanding what nanoparticles could potentially do to us. Because, with asbestos, does everybody know asbestos is bad for you, right? But when asbestos first came out, everybody didn't really understand exactly when or why asbestos could be bad for you, right? So we started learning about that after the fact. But NIEHS has been responsible for saying, well, we have these new types of compounds out that we call nanoparticles, and we're going to understand more about risk associated with exposure now, not after they're all out into the environment. So they've been putting a lot of funding in that, and we've been the benefits of that. So the other kind of compound that we've been working on is called PAHs. I'll say that to make a long story short. And these are, they're polycyclic aromatic hydrocarbons, and they're known to have endocrine disruption potential. So they're known to, what your endocrine system is. They're going to have a negative impact on that endocrine system. And they are a pollutant, so you can get exposed to those, particularly in big cities. You have more of a potential for getting exposed to these PAHs. So we did a study recently with Ricky Pereira, who's at Columbia University, and we wanted to look to see if we could determine the association between PAHs that are measured in the pregnant mother with metabolic perturbations in cord blood and health outcomes with the children later in life. Frightening news, we could, right? Because they probably wouldn't be telling you about it if we couldn't, right? Because usually science that didn't work, doesn't get talked about. But anyway, so that's what this study was about. There were many compounds that were measured that were these PAHs. There were birth outcomes measured, like the birth weight of the baby, the head circumference of the baby, even mental development indexes of how the baby's doing as it develops through life. And then we did this, what I call the birth metabolome, because I like to make up names for stuff. So that was like my new name I made up. So we have the birth metabolome, and we were able to see what we indicate here is the metabotype, and how specific metabolites or linear combinations of metabolites were associated with low birth weight or reduced head circumference or reduced learning through life. And so, so anyway, so I think I'm good, okay? So I was trying to keep over time a little bit. So those are the kind of things that we've been doing, whether they're exposures or diseases or both. And then the ultimate goal, of course, is to try to understand those metabolic pathways and what could we change about individuals to balance out your metabolic pathway, I guess I could say. So we've done a lot of studies also in kidney disease. There's a pediatric glomerular nephritis disease that's studied at Nationwide Hospital, and we've been looking at why steroids work in some children to solve pediatric glomerular nephritis, while in other children that are given the same steroids, they don't work. And this is a really important problem because the children that the steroids do not work in do not have a healthy life, and oftentimes their life is very shortened. Whereas in children where the steroids do work, they work. It solves the problem, puts them on the right track. So that's what we're trying to understand. And we're seeing major differences in these metabolic profiles between children that we call responders, who have a good response to the steroid treatment, versus those who are non-responders. So that's an example of a kidney disease study. One you may be interested in is one we've been doing with Dr. Subasagi. He is the chair of Downstate University Medical Center Dialysis Clinic. So he does dialysis. He has a large group that's what they do all day. And what they're really interested in is for people who do have chronic kidney disease, do probiotics really work? Do they make them better? Okay, so we've done a small pilot with him that was just published recently, where these were all people who have chronic kidney disease already, and Dr. Sagi really wants to know, is there a way to use probiotics to help that, right? And so we did a study where we looked at some individuals who, all the individuals had chronic kidney disease, and they all took the probiotic, okay? Got to think about that study design, okay? But some of those individuals had a good response to the probiotic, and some of them didn't. And the response that we used as the good or not good response was the level of what's called BUN, that you would, if you have a kidney disease, you would see that on your LabCorp testing, okay? It's BUN. And so for those where it got lower, that would be a good response. So some of the people taking the probiotic, it got lower. Some of the people taking the probiotic, it didn't get lower. And so we were able to use metabolic profiling to be able to understand what the biochemical perturbations were that were related to those perturbations, related to that decrease in BUN, getting better. So, and that paper's published, so you can see. I'm not doing an infomercial for the probiotic, but you can, yeah, that would be wrong. Okay, so, and then there's lots of other kind of studies. I told you about some of the work that we were doing with arsenic and PAHs, pregnancy complications, phthalates. Phthalates are endocrine disruptors. They've been related to pediatric obesity. We have a large grant funded from, who's that funded from? NICHD maybe? No, this, I can't remember. To look at this link between phthalate exposure and the development of obesity in children. We've been working with Margaret Carragus, who's at Dartmouth, chair of epidemiology there, looking at the link between metabolic perturbations and the microbiome, which is what this recruitment form that's been circulated is about. Not that I want to recruit anybody. Especially I'm not going to recruit anybody in my family, so, you know, if you're related to me, don't, you know. So that would be wrong, right? That would be coercion, right? So this is the link between why some children are more susceptible to infections, and those infections do or don't resolve with antibiotic treatment. So we're really trying to understand more about that. And we're also looking at the differences in the microbiome and the metabolome related to how the children are fed with babies. Are they breastfed or bottle fed or both fed? And how long are they bottled fed or breastfed, and those kind of things. So we're trying to understand that nutritional intake. Yeah, so there you go. much time do you have? Just a couple of minutes? Okay. Running right on time, okay? Because I hadn't had any questions. So, yeah, so I didn't know if you'd stop me or not. So I think this is the last study that I'll tell you about because this has really been a lot of fun. I met Julie Horvath because my son, who's in high school, was doing an internship at the North Carolina Museum of Sciences, and he got connected with Julie. She is the person at the North Carolina Museum of Science who's really been leading the effort to understand what our microbiome looks like, right? So if you go to the North Carolina Museum of Science, you could actually do a belly button swab with a Q-tip and give your microbiome to Julie, and she'll put it on a plate, and then she can tell you which microbiome is developing on your skin. And then she's also really interested in the underarm microbiome. So this is what's called a citizen science study. So I think it's really the new age of science now. So we're moving away from, we're always going to have, the clinical studies and the epi studies, but the citizen science is really getting the citizens involved in what they're interested in and what they want to do and what they want to know about. And that's what Julie has been spearing up for the North Carolina Museum of Science. And what we did is while Julie was obsessed with her underarm microbiome, which has been a really interesting study because she has subjects who have the microbiome of the left underarm and the right underarm, and they're different, right? And and then other people where their right and left underarm are the same, and they're wondering, like, why do the microbial populations on different parts of your body work different? Well, one of the reasons is because some people, I learned this, I don't know how I got to be my age without knowing this before, but some people sweat out of one armpit but not the other armpit or both armpits. So who knew that, right? Yeah, so I guess if it happened to you, you might know it. And then some people don't have much sweat at all, right? So you're going to have differences in your microbial population based on how you sweat, where you sweat. So that's what Julie's been working on, and then we started looking at the sweat metabotype. So this study's in progress, but where it's going to is what I'll tell you about, is that we're looking at the microbiome and the sweat metabolome to try to understand microbes and odors, and then use of personal products. So some people use deodorants, some people use antiperspirants, some people use neither. So the recruitment for this study was really interesting, that the people who came into the study came in, they gave a left arm, a right arm, a sweat swab, right? And then they got a t-shirt, and they were told to wear the t-shirt, I think it was for 4 days, right? But you couldn't wash your t-shirt, right? But you just wore this t-shirt for 4 days. And after that time, the participants came back to the museum, and they had a t-shirt smelling contest. Now you would have thought, this would have all gone wrong, right? But it all went right. Because evidently, when they were doing the t-shirt smelling contest, all you'd had was the number of the t-shirt from the different person. So you had like all the t-shirts with the number on them, and then you put how you like the smell, like it was really gross or somewhat gross or a little bit gross, or it smelled really good. And evidently, some people's sweat smells really good, right? And some people's don't smell really good. Okay? And we think that this is related to their microbial population. So what we're trying to do now is link all this together of what the microbial populations look like, and what the sweat metabolome looks like, and whether or not people have good smelling sweat or bad smelling sweat. Or even we found out that some people have good smelling sweat on one arm, but not on the other arm, right? So it's very variable. So it's just really a fun story, study, and a lot of just good basic science getting in there, like how do you do these kind of studies? And then once we really understand them, then they can be implemented in other ways too, right? So a lot of this is just leading the effort of getting people involved in projects and then understanding them. And as my last slide, I'll just talk about my group a little bit. Yuan, I mentioned, she does a lot of our mass spec. Wimal Pathmasiri, she's from Hong Kong, China, right? And Wimal Pathmasiri is from Sri Lanka, and he does a lot of the spectroscopy called NMR spectroscopy, and then also gas chromatography, time of flight. Delisha Stewart's here. She does a little bit of NMR and LC-MS, both. Reza Ganbari is from Iran, and we've been really happy to have him in the group for the past year. He came the day before the government shut down letting people come. So I was like, yay, me. I was really happy he came in. And Reza's really got an interesting project working on opium addiction and trying to understand the genetics associated with the addiction and the metabotypes associated with that addiction. Then Susan McRitchie's here, and she does a lot of that data analysis and pulling everything apart that can be crazy. And Debbie Reed, who's doing a lot of the analytical analysis in our lab. And Martin Kohlmeyer, who's been very much involved in helping me understand all of the aspects of the genotyping to try to link that to the metabotyping. So that's just some of our group, and some of them are here. And anyway, I appreciate your attention. And if you have any questions, you can ask Martin and Steve. No, it's like, you can try me first, but yeah. Yeah. Okay, thank you. Do you remember the name of the probiotic? Oh, yeah. Yeah, you can, yeah. Yeah, I can't remember. Yeah.
No, it's a real specialized
brand
for kidney disease.
Yeah. Yeah. Oh, it started with an R, but yeah, Renin. Yeah, dollars.
When you're talking about probiotics, usually these are bacteria, right?
Right.
Encapsulated and medicine world. The issue I have is there's so many different brands.
Right.
When you, as a scientist, or any other scientist, study, you select
Right.
a particular manufacturer.
Right.
They have to make certain standards. Study, and then everybody reads your study, and then publishes software codes for terms or whatever, and everybody reads it and says, okay, I'm going to do probiotics, and I can take what I buy, and they don't get the same response, or anywhere close to the same response. Also, I'm worried about the degradation of probiotics on the shelf. Because bacteria don't last very long. And if you read the label, it says 4 billion or whatever at the time of manufacture.
Yeah, yeah.
You know, so they don't give you how many are there, give you
Yep.
time of purchase. So what's your opinion?
So my opinions are probably very similar to yours. I mean, those are all concerns, and they'd be concerns in a lot of things, not just probiotics, but other kind of exploratory, even if it was a drug that's not marketed, but we're just now beginning to understand it. All the things about that drug that has to do with who made it, what else could be in that formulation that may not be the drug itself, what is its shelf life.
But usually drugs go through a process.
Right.
Whereas probiotics and dietary supplements do not go through this process of validation.
Yeah, you would be right. So yeah, and I don't disagree with you at all. That is an issue. So I think that probably, and so the reason I use drug as the example is that eventually we got processes in place to validate and understand those. And I assume that if there's enough literature that comes up with probiotics, right, that eventually we will have standardizations and processes in place to make sure that it is what it is. It is what it's on the label, and it's going to be that. But yeah.
So again, what I would say is that you're 100% right. That's what the FDA Congress cut out of this provision. But many of the diet supplements now have a USP certification.
Yes.
That means USP is quite a reputable confirmatory laboratory.
understand that. But probiotics don't meet you as a
Most of them are lactobacillus.
right, exactly.
Yeah.
Some of them make up a name for lactobacillus. But they're all lactobacillus offenders. But some of them make names up like lactobacillus rhabdomyolycus.
Yeah. This one had 3 in it, didn't it? Yeah. This one had a combination of 3, but I actually don't remember that. I didn't look it up because I didn't want to do an infomercial for the company. So I didn't, yeah. Yeah, sure.
Back you were talking about animal models and your research with metabolomics or metabolomics, however you say it. The issue comes up is, in the beginning you were talking about how specific this particular situation with metabolomics is for humans and for individuals, for people. And then you're talking about an animal model. And I don't see how the two
Okay.
because an animal model wouldn't have the same biological,
Yeah, I got it. Gotcha. Okay, so so it's really great because our metabolites are greatly conserved across species, and so what we detect from a tissue, whether it's a biological fluid or an organ tissue from an animal model, is very, very similar to what we're going to detect in a biological tissue and fluid from a human, because those metabolites are greatly conserved. Now, what's different is that the flux or the movement through the metabolic pathways differs. So we have, there's 115 mammalian pathways. And so pretty much they're conserved across species, not totally. I can think of one that's associated with scurvy, right? The synthesis of vitamin C that's different between a rat and a man. But typically they're very well conserved across species. But the amount of the analyte that's detected can be different, right? And so the ratios of those metabolites on the pathways. So this is why it becomes so important that when we're talking about our animal models and we're interpreting that data, we interpret it in the context of the metabolic pathway, because even if the specific analyte might not be the same that's being increased or decreased, that metabolic pathway is what you would focus on for the increase or decrease. So it works really well. Unlike some other technologies, I think, where, like for genomics, you may have to have specific gene chips for species and all. But the beauty of metabolomics is once you have your assay set up, you can use it on anything. It's really nice. Yeah.
Metabolic pathways the same or they different?
Yeah. So this is a great question. So we think if we looked in biochemistry textbooks, we have so many metabolic pathways, and these are our mammalian metabolic pathways, right? But sort of the point of what we're trying to do is to understand what those individual differences are. So you can have differences in those pathways because, for example, you may be missing a SNP, right? Or or a genetic component may be different in this person versus that person, and so that's going to give individuality. And then also there can be differences because you may be taking a drug that interacts in one metabolic pathway, so it's keeping it busy in one way and not the normal way, right? So if you compared that with another person, you would see differences there. So, or chemical exposure. So there's all these things that just happen to us all day, even eating food. So even if you're drinking tea or coffee or water or orange juice, you're changing that balance of pathways from one person to another.
So, to make sure I understand step one.
Because we're going to have the test. The test is coming up, yeah.
So, in the study of your metabolomics, that is an expression of our genetic makeup. So that, for instance, when you studied the pregnant women that had
Placental abruption, yeah. PA.
PA, we couldn't know from their genetic makeup that they were going to have a PA, but we found out from their expression with how they metabolize that there was a chance that that was going to happen. But you couldn't say because of, when you say coming down here, we're really confused about genetics.
Right.
And I'm trying to make a leap. Like, I understand that with opioid addiction, that if you are a rapid metabolizer or if you're a slow metabolizer, their propensity for opiate addiction increases.
Mm-hmm.
But is that right?
You could be.
Studying what is the expression of our genetics, we all don't have the same metabolic pathway.
We pretty much have the same metabolic. There's good metabolic convergence, but it is related to genetics. But if you took placental abruption, for example, no one has ever been able to find a genetic reason for placental abruption. However, higher rates of placental abruption, Delisha, I'm correct, occur in African American populations, right? That's my health disparities expert, right? I think it's higher in African American populations. So you would think there's probably a genetic component to it. So there's probably a lot of genetic influence, but then we're looking at other things that could happen after the genetics. Like you could have stress or chemical or drug-induced post-translational modifications on proteins that could change your metabotype also. So I would say it's the sum of your exposures, whether or not those exposures are your genotype, which you start with, right? Or then all these other exposures you can get that happen, like whether you get BPA in your drinking water or, whatever else is happening in your life, right? Yeah. So it's a combination of these factors. And that's actually one of the reasons why NIEHS has been started up this new program funding Exposome, because everybody's realizing that, genetics, maybe I shouldn't use the word gun recently because we can't do that anymore, but I heard a director of one of the NIH agencies say this years ago that genetics loads the gun and the environment pulls the trigger, which is sort of interesting because you have your genetics, which says what your risks are going to be, right? And then you have something that triggers those, okay? So, yeah. Oh. There you go. Yeah. Right. Right. Probably so. And so we are working, so we have a team that's been pulled together from the National Institute of Drug Abuse, the National Cancer Institute, and then our group at UNC to understand the genotyping and the metabotyping, but there's also a microbiome component that's going on within those studies also that I didn't mention. And so we do think that if you're exposed to the drugs, it probably does modulate your microbiome, which probably does correspond with maybe even your addiction profile is what I say, because you have some people that are mildly addicted or moderately or strongly. So the study that we're doing now is really focused on the genotyping and the metabotyping of individuals, but then we're also exploring how we can include the microbiome in there. So it would be a really new conceptual framework. Yeah. Yeah. Mm-hmm. Yes. Yeah, so I think that's the opioid crisis is the reason that NIH did form this team to start working on this, and we've actually partnered with a group in Iran who has a really good cohort, a good population of people that are exposed to opium. So in the work that we're doing, some people are opium users, some people are nicotine users, tobacco product users, some people use both, and some people use neither. And we have a good sampling of male and female from that population, and we're trying to make this link now. So that was a study that just got funded last year, and why Reza came from Iran. So So one of the, yeah. Yeah, and I don't know that much about alcohol. Our alcohol experts aren't here, but Susan Smith and Phil May. But NRI does have 2 groups here that are working on alcohol exposure and outcomes in children. It's not as much on the addiction profile as the outcomes in the children, but the alcohol addiction is really important. Yeah. So. Steve, would you like to take? Steve is the world's expert in choline, so he gets to answer that. Yeah. That's what I eat. Yeah, I started eating 2 eggs a day after I started reading about choline. Seriously, like. Yeah. And I've been told to thank you for attending. We really appreciate that.